talk about today. Now, genome engineering is something which people had in mind for quite a long time. And there are 4 examples here. The meganucleases, zinc fingers, talons, and CRISPR Cas. Now these 3 here are based on nucleases which are transported to specific sites on chromosome with certain proteins here. With these blue binding sites, for example, zinc finger binding sites, and this is a meganuclease. And in fact, I mentioned a company here, Sangamo, which has a compound in clinical phase 2, I think, using talons and trying to edit genes. And it's an interesting, it's phase 2, so we don't know what's going to happen, but we'll see. So, but the hype is very much on CRISPR-Cas. These are almost forgotten. Not quite. There are some, also these talons, transcription activator type compounds are still used in the plant field, as we will see, because they started the work 5 or 6 years ago when CRISPR-Cas did not exist. So there are some experiments, and even on the market there are some plants which I will come to, which are using this particular system. But the others are rare, and but for the sake of completeness, I have introduced them here, and they are often summarized as in this paper, which I mentioned here. This is a group in Beijing, Kaxia Gao, at the academy, where, and she's really using this intensively to modify crop plants. I have an example of that later. So what is being done? This is a very recent paper, I think. No, not a very recent, but a paper which summarizes everything you can do with CRISPR-Cas. You can make deletions, insertions, replacement, DNA modifications, even you can add methylene acetyl groups, as we will see. So you can really do almost everything: transcriptional modulation, RNA targeting, and applications in basic research and human medicine. We heard human medicine just a minute ago, and agriculture, biotechnology. I mean, everything is now amenable to using this technology. Now, to understand what's going on, I think it's important that we think a little bit further. This is the mechanism of this is the traditional locus where with the Cas genes and with these repeats and spacers, the CRISPR repeats, which are transcribed into the CRISPR RNA. And then there is this second RNA transcribed to the left here, which is absolutely necessary to this red to bind to the CRISPR RNA, and then there has to be this protospacer and the PAM sequence, which I mentioned yesterday, and then the DNA is cut. So the idea is to create a double-stranded break, and most discussions really focus on how to create this double-stranded break. But the applications of they go on from here. They start actually here. What happens with the double-stranded break? Can you repair this somehow? Can you add new nucleic acid sequences and these kind of things? And and in order to do this, you have to start with a double-stranded break, which is now created by CRISPR-Cas or some of these other mechanisms, and then you have 2 ways of dealing with this. You can correct this by non-homologous recombination. Then you introduce very small stretches of DNA, you digest this maybe a little bit, and you create something which are called Int'l mutations, insertions and deletions, just a couple of nucleotides, 2 or 3. Then you have a mutation, a knockout in most cases. But you can also have homologous recombination, and in order to distinguish between the two and to guide yourself into what you want to do, in this case you have a donor template which overlaps here the region of the break, and then you get a precise insertion of a whatever sequence you offer here, the donor sequence, and that this precise gene editing works very well. There are now papers where you have stretches of almost 10 million base pairs or so, really long stretches you can introduce this way. I mean, people are talking about introducing humanizing stem cells from pigs, for example, and making them compatible for the human major histocompatibility complex, which is, of course, vast stretches of DNA. And that, in fact, has already been done. So offering the system these donor templates really directs it into a what they call precise gene editing, and creating almost completely new genomes. On the other hand, you can create knockouts, for example, and knock-ins by just not offering anything, just letting it produce the double-stranded break and then correct itself, and then you get these small mutations, mainly knockouts. Now, examples of this, of course, are, since 2 years, are relevant. The first one, maybe in human cells, is a lentiviral construct which is used, which you can buy at Adrien and other places. And I have described this is from the group of Feng Zhang at MIT. The first experiment actually with human cells. These are 293 cells which have been used here, a kind of HeLa cells, as you know. And they had a GFP gene inserted them, and using this construct here and the Cas9 cassette and the guide RNA in red here, they tried to knock out the endogenous GFP gene, and that happened in the yield of 93% of the cells. As you can see here, almost all of the EGFP fluorescence has disappeared, while if you use a single guide RNA which is not specific, then you get kind of incomplete destruction of the particular gene. This is a construct which lots of people are now using in human system or in mammalian system, very efficient. As I said, 93% of the cells, I think. It's described here in details what they all do to have it efficiently working. Furomycin markers, packaging signals, response elements, so everything you kind of need to do it in mammalian cells. But things also work in plant cells, as I mentioned before. And maybe I decided to add 2 examples of these knockouts, these non-homologous recombination examples for plant systems. Now, plant systems are complicated. For example, this is wheat. Wheat is hexaploid, so in each it has 3 genomes or 6 genomes, and the genomes are more or less homologous. They are not 100% homologous. They are about 93% homologous. So you have to find an exon which is fully conserved, or you have to adapt a little bit and find guide RNAs which are really complete. And that can be done quite easily. As is shown here, this is an example from the wheat genome editing. There is a problem which the people who, like all of us, live with. This is mildew, which is the most terrible disease in wheat. And you have to, it's a fungus, and you have to create resistance to it. And that has been achieved. You need, I think I have a good example here. I have a good example here from these papers. So you have the 3 genes here, MLO genes they are called, and they are all used. They have a guide RNA which is in exon 2 here, and then the 3 genes are knocked out, and they can, using PCR reactions and so forth, they can check and see, here it is, that in cases where only 2 of the genomes are knocked out, they are still sensitive to the fungus. When the 1 out, they are still, but when all the 3 copies are knocked out, then the wheat is resistant. So, and this is the same thing here. The wild type, which is affected, and the mutant one, which survives, and that is in fact already grown on land of the Chinese Academy of Science in China, and that creates, by the way, an interesting problem. But before I say, I have summarized a few genes which are used already in this connection with crop plants, so very different stories than the one you have seen before. For example, in corn, they are working with phytate biosynthesis. Phytate, you know, phytic acid is a sugar actually. It's an inositol hexaphosphate, which binds all the phosphate which is present in our organisms, and we cannot digest it at all, or practically none. It's all so, but we need lots of phosphate, so you have to get rid of this to some extent. And then there are a few other examples, which you see here for soybean, rice, and wheat. The wheat example on the bottom I've already explained. There is this fungus called Blummeria graminis, which none of you probably has ever heard of, but people who like to work with wheat, and I think wheat is the 3rd biggest crop in the world, about 700,000 tons per year, is so much affected that in Canada, which is a big wheat producer, at the moment about 40% of the harvest is destroyed every year, so they have to really get going. And so the CRISPR has really introduced a new perspective here. The problem is, and that raises a question which you have all heard about. The problem is, this is the cover of Newsweek of last May, sometime last May. There is a problem now that whether this is a GMO or not. And as you know, in Europe there are no GMOs plants, and they are forbidden in almost every country in Europe, even though people are very much afraid of GMOs, but they don't exist. There's not a single permission for anything. And the problem here is that there's no trace of the CRISPR-Cas visible in these plants after they have been modified. So they look identical to any other plant which has been modified by X-rays or by chemicals, as the seed industry usually does. They usually accelerate mutation rates by doing chemistry or radiation. And so there's no trace left. So you cannot really see whether the organism which you have created is a GMO or not. Now in the US, the system is different. In the US, the system was always looked at by looking at the product and not at the process. While in Europe, we didn't care about whether the product was good or not, we just looked at the process. And then since the process was a GMO process, it was not permitted, especially of course that in the good old days of the old recombinant DNA debate, they always had antibiotic resistance genes or things which were which were necessary to detect the transformed clones. Now the system is so efficient, as I have shown. I have shown you the data that they don't need any antibiotic resistance or other markers, so they can really do it like this very efficiently, and therefore this question is now in front of the authorities in Brussels, and we will see. Of course, the Greenpeace and others don't really like these new mechanisms because they cannot really show that things have been transformed and are GMOs. And therefore, if you cannot show anything, it's very difficult to make legislation against it. We will see the outcome, but lobbyism is really very strong and is very rarely rational. Now, in this case, I think I'm not sure what they do in Brussels, but there's a strong lobby, is stronger than us. And so it has to do with these ethical issues to which I come in a minute. So that is what was published in 2014 in the famous Doudna and Charpentier paper, the model organisms which had been used already at the time. You see, I mentioned the 293 cells, which have the humans. These are human embryonic kidney cells, and many others. I think now you could really make this list. I think anything which is possible has been tried, and so it's a very general. So what can they do? So you can adapt the system. You can adapt to study transcriptional activation and transcriptional repression. I'll mention that in a minute. You can make epigenetics by fusing a protein, by fusing enzymes which acetylate chromatin or DNA, and then you have an acetylated, and you can make epigenetics. And the same you can do that with methylases, so you can add methyl groups. You can map enhancers. You can create animal models very easily, and you can make so-called gene drives. I come to that in a minute, and I will. This is the applications of a catalytically inactive Cas9. The group at MIT, which I mentioned already, has created a so-called DCAS, which is the dead Cas, which is a mutation in one of the, I think it's 1008, a histidine against alanine, and that Cas9 is dead. Binds all the single-stranded RNAs and everything, but doesn't cut anymore. So it doesn't have nuclease activity anymore. So it's called DCAS. And this is what I have described here. And now you can fuse to the DCAS protein. You can fuse, for example, transcriptional activators, VP16, which is a domain, which is a very strong transcriptional activator, as some of you will know. So we have now a fusion protein which binds DNA wherever you want to bind it, using the guide RNA at any position where you want to bind it, but it doesn't cut, but it binds. And then because you have a transcriptional activator, transcription proceeds very active, so you can activate any gene which you want anytime in a given cell. And the same is you can repress a gene. This is CRAP, is the cripple gene is a Cripple is one of these development genes in Drosophila in the early development, and it has a transcriptional repressor, which is probably one of the best ones around. And you can fuse that domain, and then you get what they call heterochromatinization. Yeah, then you stop the activity of a particular region. I think we heard earlier this morning how this initiation process runs anyway, so you can stop it at any given place. So that is really quite useful, and has been used, and there is some literature here which I don't want to go into. It just so the concept is using a dead Cas, a mutated Cas, which is also commercially available, and that mutated Cas, of course, can be linked, fused to various interesting proteins, And you can already see that you can go even further and add acetylases or methylases, and then anything goes, so to speak. Now, a very good way of dealing is to create transgenic animals. I think, Glauco, that is what they do in Monte Rotondo. I'm sure that it used to be very difficult to make transgenic mice, because you had to be back crosses and everything. Here you just use either you use a zygote, inject the target sequence together with Cas9, and then make an embryo transfer and get the mutant mouse. I think it takes just a couple of weeks until you have a mutant mouse to produce a mutant mouse. That was the first paper, I think, Janisch in 2013. This, again, the MIT group at the Whitehead who did this. And you can also do this with embryonic stem cells, which are used anyway in this system, and create, again, almost any transgenic mice which you want to have. And that is also works for other animals. It works for worms, and we come to the question whether it works in people, in us. I come to after the coffee break. Now, before the coffee break, I want to introduce another application which is quite interesting and threatening almost, and that is gene drive. It was published last August or so as a scheme outlining, as a paper describing the so-called mutagenic chain reaction. And it can convert heterozygotes to homozygous mutation immediately. You know how to do this. Normally you have to make crosses, and then Gregor Mendel applies, the Mendel's laws applies, and then you have to make back crosses, F2 generations, and then you get from 1 to 4 to 2 to 2, and then eventually you get purebred homozygous mutations. Now here you can do this much easier, as it's described here. You have a genome and have a gene which you want to mutate, which is a heterozygote, which is heterozygote mutations. Now, using Cas9 and guide RNA, you introduce this vector. You take this vector construction, add it to a system which has been a double-stranded break here, and then, of course, it is introduced into the genome, and suddenly you have a genome which has that new construction, Cas9 in this chromosome within this particular gene, and then you make homology-directed repair. This will be copied, of course, and then you suddenly have, within the nucleus, without any breeding, you have 2 chromosomes which are affected. So you have, you start with a heterozygote which has only 1 copy, suddenly you have 2 copies. Within a couple of days, probably. It depends on what kind of organisms. And now you can see it here on an experiment. This was done with flies. You can see this is the yellow locus. And you can see here the Mendelian inheritance. Of course, if you have female and males, you get, this is a heterozygote, so they are all positive, and they would be negative. There would be a knockout if both chromosomes would be affected. Now, using this scheme, which I've just described, this MCR gene, you can destroy any given gene in Z2, and then you suddenly have already something which you only normally achieve after back crosses. You know, this you can, of course, back cross, and then you have these 2, and then you get 50-50, but it takes a while. It takes F2, and here you get it right away. And you can see here the ones with the knockout and the ones with the knock-in, and it works perfectly. So you can immediately transfer a gene throughout an entire population. if you add the Cas9 and the guide RNA into their chromosomes, into a particular genomes, into their genomes. Now this is, The Economist talked about bye-bye Brother Mendel. You may have seen The Economist edition, which describes it again here. And yeah, you see here, this is what I just tried to describe. Make a double-stranded cut, repair it with Cas9 and the guide RNA which you want, and then you make a mosquito. You do this in mosquito. So in a single experiment, almost all the offsprings are mutated. And this is not true for mosquitoes only, it also works in yeast. Actually, one of the first experiments this is why yesterday I had a photo of George Church, was done in yeast, where you can have a Mendelian inheritance using the adeno2 marker. Is a wonderful marker which maybe you have used in the course, where this is a nucleic acid, a gene in nucleic acid biosynthesis, which turns red or not, and so you make the cross here, and then you get a 50-50 inheritance. But with a gene drive, you get a diploid which is already positive in both chromosomes, so it's homozygous. It started heterozygous, and then it was made homozygous by the addition of the CRISPR and the drive, and then you get a complete non-Mendelian inheritance, and this describes again the process of homologous. Now this has raised a lot of criticism because in fact people are thinking of using it in Panama and other places to eliminate mosquitoes which transfer malaria or West Nile fever or something like this. But there are problems. If you look at this, there's now a literature, rather recent, I think November of last year, describes what to do in the lab if you want to do experiments of this type. Because the problem is you don't want these mosquitoes to be released, because how do you get it back? The gene. Maybe you don't want the gene because it has a phenotype which has problems. You all know very well, and we come back that when I talk about the human applications, there's always problems which you cannot predict. For example, would you ever predict that people who have sickle cell anemia are resistant to malaria? Now, so there's something else going on always, and that could happen here too. If you knock out a gene which creates sterility in mosquitoes, then it could do something else to them which you don't really want. Suddenly they transfer some other disease, some other virus, or some other parasite which you don't want, and therefore there is laboratory strains which cannot reproduce, and you only release in areas where there are no wild mates, for example, and there are physical barriers and all kinds of things which you can do and which you have to do now. It's already in the books which you have to do if you want to do gene drive experiments, and I think it shows that how CRISPR-Cas really revolutionizes the entire field of genetics. I think with that, I have a few publications here which are relevant, which I mentioned, and the first one relates to the human embryo engineering alarm, and that is what Glauco asked me to talk to you after the coffee break for a couple of times. We can discuss here among each other what should one do with people in this context, and I think is that the right time? Yeah. Thank you.
Thank you.